Are we ready? We're recording.
Right now.
It dinged.
It's going to look embarrassing at the start on YouTube when people are looking.
Mm-hmm.
Oh no. Okay. So last time we spent the time talking about the different families of glucose transporters and the regulation of the glucose transporters in muscle cells. So now we're going to assume that, oh yeah, let's make as much noise as we can to start the video.
Jeez, Ellen.
I think Ellen wrote me ready to start at the start of the hour rule.
I hope he starts this year.
So now we've got glucose transported in. So in whatever cell type, glucose is now inside the cell. And so now the first reaction that we normally associate with glycolysis is the phosphorylation of that glucose by a kinase, either hexokinase, which is active in all tissues, or glucokinase, which is an isoenzyme of hexokinase, but that is present only in the liver, to make glucose 6-phosphate. Okay? Now, I alluded to this at the end of the last class, which is going to confuse anyone not from this class who watches this video, because that class video is not available. But, all right, I alluded to this. What is the purpose of this phosphorylation reaction?
To trap
glucose.
Now, by phosphorylating it, the glucose 6-phosphate is not a substrate for the glucose transporter family. So once you've phosphorylated it, the glucose can't get out of the cell anymore. Right? So you've trapped it in the cell. There's one thing.
Keeps the concentration low.
Yeah. If we look at this equilibrium, remembering that all reactions are equilibrium reactions, if we're hydrolyzing ATP in the process, where is the equilibrium of this process going to lie? Very far this way. So what happens then to the concentration of free glucose inside the cell? It's kept low by this reaction. So what does that do to this gradient here? It keeps it flowing in. So even though we transport this in, and we say we don't use an active transport process because we don't use ATP or another metabolic energy source in the direct transport, inside the cell is using ATP energy to drive this reaction this way and to maintain as much as possible a gradient of glucose concentration across the cell to keep flux of glucose in this direction. Okay? So that's sort of the purpose, the first purpose of this reaction. Okay?
Would that be considered secondary active transport then?
It's not, right? Secondary active transport is when you use a gradient created using energy as your source. This is sort of just an indirect way of influencing transport using ATP inside the cell. Okay? Now, back to this difference, hexokinase or glucokinase. That is, hexokinase working in all tissues, glucokinase specifically found in the liver, and again, whenever I say something is specifically in the liver, what do we say in the back of our minds? No? It's going on in the liver, it's also going on in the kidney, but the kidney quantitatively is so much less important, right? The kidney is like the liver's little brother back there, does the same stuff, wants to follow around, but it isn't big enough to really make any difference. So the kidney, at least certain portions of the kidney, tend to do the same things that the liver does, just out of relationship. Okay, so if we look at hexokinase and glucokinase, so this is summarized here, and then we'll talk about it here. Bless you. We'll talk about it again with a picture. So hexokinase, Km for glucose of 0.1 millimole, approximately, right? If you look at hexokinase across species, that's the range, plus or minus a little bit, that we get for the Km of hexokinase. Okay, so let's think about that. What did we say the concentration of blood glucose is? 5 millimolar. Okay, how does 5 millimolar relate to 0.1 millimolar? It's a lot bigger. How much bigger, Devin?
50 times.
50 times. So the concentration of blood glucose, normal fasting blood glucose, is 50 times the Km for the hexokinase enzyme. And if we assume that this is trying to equilibrate, right? So that this is flowing in, this is going to be something lower than 5 millimolar, but still a lot higher than 0.1, right? Because it's constantly flowing in. What does that tell us about the hexokinase enzyme?
Probably saturated.
Probably saturated. Yeah, that's probably a good guess, right? Now, the other thing about hexokinase is that hexokinase is inhibited by its product, glucose 6-phosphate. Right? Glucose 6-phosphate inhibits hexokinase activity. Does that make sense? Robbie, why does that make sense? Robbie's going to stop nodding at me because I always call on him when he nods at me.
Because I think if you have too much, it'll stop the catalysis.
If we get too much glucose 6-phosphate, it will stop the catalysis. If we get to, what are the situations where we would get too much glucose 6-phosphate?
There's a lot in your blood.
What are we going to do with the glucose 6-phosphate? Elizabeth, what are we going to do? Right, but in general, where do we want that glucose 6-phosphate to go? Well, what's the whole point of our lecture today? Glycolysis. We want it to, right? We want it to go through glycolysis. So if glycolysis is working and active, when we make this glucose 6-phosphate, glycolysis metabolizes it. So what's one time that glucose 6-phosphate might build up? If we're not using it, right? We only do glycolysis when we either need quick energy, and the liver will do it a little bit to just get rid of extra glucose, right? Take it down and make fat out of it. But most of the time, in the muscle, for instance, we only do glycolysis, really, when we're trying to make energy. Me pacing back and forth here, I'm doing glycolysis because I'm not doing an aerobic activity, right? I'm not doing it enough to transition my muscles to an aerobic activity. I'm just pacing back and forth. So I'm doing this, taking it through glycolysis. But if you're at rest, not going through glycolysis, you make glucose 6-phosphate, there's no place for it to go, and so it feeds back and says, hey, let's not do this anymore, let's not build up a ton of this that we don't have anything to do with it. Okay? So,
Did you say what kind of inhibitor it is? A feedback?
no, I'm not sure. I think it's allosteric, but I'm
That's
not
fine.
sure. I actually just spent the last hour looking up, I was trying to find the mechanism of hexokinase and glucokinase. I'm just curious about the enzyme mechanism. Your book has something about the mechanism, but it's non-specific about the amino acids involved. And I wanted to be more specific, but I couldn't find the information I was looking for. Okay? So I think it's allosteric, but I'm not sure. I think it's got a second interaction site. But I could be wrong. Somebody on YouTube will probably correct me. So that's advantage or disadvantage of going public with these
Do
things.
people actually correct him?
What?
Do people actually correct you on these videos?
I haven't been yet, but I don't screw up openly. So, people, I'm sure somebody will find it if I screw up. And since I screw up regularly, I just don't usually screw up where,
You're on
billions of people can check me. Okay.
So it's the trillions going viral.
Right? There's real pressure here for me.
You're not on camera. It's okay. It's just your voice.
Just my voice. I was on camera last time, though. And it's my YouTube account. Okay,
Yeah,
so that makes sense in terms of what the biological system is trying to do, right? Inhibition so that this doesn't build up. The other time we would get high concentrations of glucose 6-phosphate is if we transition and we're talking about a situation where we are doing gluconeogenesis or breaking down glycogen. So again, let's go to a muscle cell. So a muscle cell is breaking down its glycogen, getting hexose phosphates from the breakdown of glycogen to go through glycolysis. If it's actively breaking down its glycogen, it might not need to bring in glucose from the bloodstream. So again, that glucose 6-phosphate could inhibit this so the muscle could work on its own glycogen stores, not glucose coming in from the outside, as a way of conserving glucose in the bloodstream. For what? What uses glucose in the bloodstream? Your brain, yeah, right? As a way of conserving blood glucose for use by the nervous system. Okay? So, those are the situations when we might have other ways of making this. Now, what if we're in the liver? What is glucokinase? Now, first, with glucokinase, we can't talk specifically about a Km. We talk about an S at which it is half saturated, a substrate concentration at which the enzyme is half saturated. Now, that substrate concentration at which it is half saturated is about 5 millimolar, right? So, note, suddenly we're in the, halfway up the curve. Now, the reason we have to call this a substrate concentration at which it is half saturated and not a true Km is that glucokinase has sigmoidal kinetics. So its enzyme kinetic curve has an S shape like the hemoglobin oxygen binding curve, meaning it is positively cooperative, right? It exhibits cooperativity.